Oke. Selamat pagi Bapak dan Ibu dan teman-teman. Pagi ini kita berbahagia karena kita akan mengikuti seminar dari guest lecture. So good morning everybody. We are glad today because we have a guest lecture from Japan, Professor Teruo Sone, Ph.D. Thank you so much for coming to our place. He is from Hokkaido University and Bu Hagia supervisor. Where is Bu Hagia? Thank you for Bu Hagia to bring your professor here. So, and next. And also we are live on YouTube. Hello, everybody on YouTube. And also on Zoom. They are joining us in Zoom this morning. Thank you for joining us today. And here's our guest lecture CV. Now, Professor Teruo is Dean of Graduate School of Global Food Resources at Hokkaido University. And his research area is applied microbiology and molecular plant microbe interaction. So we know that some of you are master student and PhD student. If you are interested to go to Japan, so just see his research area. And okay, now he is in Center of Education and Research for Hokkaido Wines, Hokkaido University, and at Laboratory of Applied Molecular Microbiology. And so you have two laboratories. Yes. Oh, laboratory for Novel Pagu of Hokkaido Wines under Research Faculty of Agriculture, Hokkaido University. So he has an email also if someone interesting to contact him. Okay, and today we will hear the seminar with the titles. What it is? Mutation in rice blast fungus, host specificity and fungicide resistance. And I think it's a very interesting topic for us. So we write that student from pesticide in plant protection and student from pathogenesis and host response must attend this seminar because it's related to our topic in the class. Yeah. Okay. So we have from 8 to 10 o'clock. So it's depend on you how long it will be. But of course, it's open for question from a student from Zoom, from YouTube, so we can discuss it. So, and then time is yours.
Okay. Selamat pagi. おはようございます。Good morning. Thank you for your kind introduction. I'm Sone from Hokkaido University. I'm very happy to be here because actually this is third time to visit Bogor. But last time I was here was I think 2004, 2005. So almost 20 years after the last visit, and I'm seeing the so changed the Bogor. I was so surprised, and this university campus was so beautiful. So I was I'm very happy, and I'm another surprising for me is that you have the lecture in this early in the morning. It is unbelievable from Japanese. So one of Japanese students there, but we usually start on 8:45. Even from 8:45, some of the students cannot come. Yeah, so wow. Okay. So so very surprised. Yeah. Okay. Anyway, let's start my lecture by sharing my slides. Okay. Yes. So today I would like to talk about the rice blast fungus. As in introduction, I'm now working in wine too, but I start the research in rice blast fungus when I was student. So the length of the time or the history of the research, rice blast fungus is the longest, the most like the long history for me. And all the time I'm studying about the rice blast fungus, that was about mutations. Okay, so today I will talk. I'm expecting to something like less than 1 hour about the mutations of rice blast fungus. In one topic is host specificity. So host cultivar specificity. Yeah. So in other words, maybe you know the pathogenic race. So they overcome the resistant cultivars. The other topic is fungicide resistance. So fungicide resistance is very important, and Hagia is also doing about the fungicide resistance now. But today's topic is a little bit different from the just become resistance, but it's how to spread the resistance to the population. Okay, so I'm going to start with the introduction of rice blast disease, but maybe all of you know about the rice blast disease. So rice blast is the most important or most damaging, devastating disease of paddy rice. In Japan, we have the half of disease loss due to rice blast. So most important disease. And this tendency is almost same in the world. So rice blast is the most important. And what the symptom is that it will make a blast first in the leaf, on the leaf, and then that symptom will coming up and up. So if it's infects on the panicle, it will make much loss of the harvest. So this is the photo showing the I took in the Hokkaido Prefecture Agricultural Research Center. So this is not the natural, half natural maybe, because they put some infected rice in the center of here. And after 1 month, it will become like this. So it's the disease was spreading. So if you find some symptoms like this on the paddy field, it might be the rice blast. So the pathogen of the rice blast is rice blast fungus. It's fungus, Ascomyces. Okay. The name of the fungus is Teruomorphis Magnopose oryzae and Anamorphis Picularia oryzae. So, but only Anamorph is found in the field. So we have the Teruomorph or perfect stage in the laboratory condition by using some specific strain. We can see the perfect stage, but usually if we isolate the rice blast fungus from field, those are only those will show just only Anamorph. So their life is start from conidia, and it attaches to the surface of the leaf. It will germinate they need a humidity, and they will make a germination, and they will produce a specific cell, a presorium, and this circular or bulbous cell, a presorium, And this upper sorghum will accumulate the glycerol inside. And please remember, this is happening in the water drop. So by the accumulation of glycerol, water will coming inside this upper sorghum cell. And this upper sorghum cell is very strongly attached to the leaf surface. So the pressure will going up inside the cell. We call target pressure. So that using that pressure, the rice blast will penetrate into the rice. Okay, so after the penetration, they will expand their hyphae inside the cell and make another sporulation or make another conidia to do another cycle. So this is a life cycle of them on the rice. So actually, this is the same slide as previous, but how we are controlling this disease. One is agrochemicals or pesticides, fungicide, and the other is resistant cultivars. So we're using these two methods to control this disease. So I'm going to about the topics of mutation from now. First topic is a mutation in host cultivar specificity. In other words, mechanism of ABR gene mutation. So ABR gene is the gene controlling the host cultivar specificity. So this is the symptom on the leaf of the rice blast. So you may see this is the successful infection, result of successful infection. So they start infection in the center of the lesion and they will expand. So in the beginning, in case of successful infection, if we make the cell as transparent and observe under the microscope, this circular thing, this is the upper sorghum that's made. So from upper sorghum, you can see the hyphae is expanding to another cell, another cell, another cell. So finally they will become like this. But if this rice blast fungus start to infect on the resistant cultivar, the rice will turn on the system of the resistance. Okay, so after such resistance, the fungi will die together with the host cell. So they will kill the fungi when they start the infection. So from the point, if you see using just naked eye on the leaf, you cannot see the big lesion, or sometimes very small, tiny lesion you can see, but that will be a symptom of resistance. Okay, so this resistance is controlled by two factors. So one is R gene, resistance gene in the rice genome. Okay, and the other is the factor or gene in the pathogen, ABR gene. So even though the plant has R gene, if the fungus doesn't have ABR gene, they cannot turn on the resistance. Only when the pathogen has ABR gene, it will show resistance. So it's a combination between the rice gene, R gene, and ABR gene in the rice blast fungus. So today's topic is the mutation of host specificity. So which means if the rice blast fungus makes mutation in this ABR gene, resistant plant, I mean a resistant rice cultivar, will not show the resistance. Okay, so from this part to this part, it will change. Okay, so the problem I'm talking about is that the mutation in this ABR gene will make the rice plus fungus to gain the pathogenicity to the previously resistant cultivar. Okay, so I'm talking, after this, I'm talking about the mutation of ABR gene. So this diagram or this figure, I took from kind of the textbook, that this is a general understanding of the how we doing against the change of the ABR gene or the breakdown of the resistance. So, okay, so let's start from here. Okay, if we human found the new R gene, the new R gene will be used for the in the field. So that will be showed effective control first, because the most of the rice blast pathogen population has the ABR gene. Okay, but at the same time, that will increase a selective pressure for pathogen which overcome the R gene. So if they, that condition, if there will be a mutation in the ABR gene of the fungus, that condition will be very favorable for them. Okay, so that under that pressure, somehow the new pathotype or the mutation in ABR gene will occur, and that resistant cultivar will be broken down. Okay, so we call that breakdown of our gene. And then we should look for the new R gene and do the same thing. So as a result, so first the introduction of first R gene, resistance first showing, and it's broken down. And again, new one broken down, another one also broken down. So this is a cycle of the breakdown cycle of our gene, we call. It usually happen in 8 to 12 years of cycle. And actually in the rice of Japan show this cycle. So what I'm thinking for these 20 years, I'm doing more than 20 years, but is if we can know how they make mutation in ABR gene or mechanism of mutation of ABR gene, maybe we can control the mutation itself or just anticipate when the mutation will occur. And then we can prepare for that mutation or change of ABR gene. Okay, so I was very interested in the mutation in ABR genes. Today, I'm going to talk about the mutation in ABR gene, but what was difficult was to clone or to know what is ABR gene. I started the work in 1992. And in 1994, I was writing into my laboratory notebook that to clone the ABR gene. But finally, we could clone in 2009. So it took 15 years to clone only one gene. But today I'm going to talk just after the cloning. But okay, why it was difficult is the ABR gene is, if we want to clone some gene of known protein, it will be not difficult because we know the sequence of the protein, amino acid sequence. We can infer what kind of protein will be there. But for the ABR gene, we didn't know anything. So what protein will be encoded? Nobody knows. So at the time, what we could do is using genetics. So making the cross between two fungi and making genetic map and the map-based cloning was the major way. So okay, yeah, that it takes time. Okay, so after the cloning, many research was done for what kind of mutation happening in the ABR genes. So this slide telling about one of the ABR gene, ABR PITA, that's corresponding to our gene PITA, in 2010. Okay, so this is not from our group, but anyway, so this is the illustration of the gene locus. So actual gene itself is this white part. And this is, so from here to here is the gene. So intact gene has a promoter and the gene. Okay, but from the field isolates, it was found that there will happen the base shift to make frame shift to stop the translation, or deletion of the 5' region, or deletion of all genes, or sometimes insertion of the transposable element, or simply base substitution. Okay, so many different type of ABR gene, mutations may happen in ABR gene. Here, I will talk a little bit about the transposable elements in rice grass fungus. I don't want to make you to memorize the name, but please understand that rice grass fungus has so many different kinds of the transposable elements. This is name. This is the name of transposable elements. And it has very different type. So retrotransposon, which is related to the retrovirus, which requires the reverse transcription, has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different kinds. And DNA transposon, at least four kinds. I think there will be more different kinds because after we have the genome sequence, we realize they have so many. So researchers gave up to name each one. Yeah, actually, it's very that's a kind of problem. I will talk about it later, but yes, so many different kinds of transposable element. That is one of the key of the mutation. Okay, so another type of the ABR gene I will introduce, ABR PIA. This ABR gene was cloned by our group. So it's 2010. I started that in 1994. So, and this is the result of the sudden hybridization. Okay, using 18 different field isolates. And those field isolates showing the pathogenicity against PIA resistance gene. Okay, ABR PIA is ABR gene that's corresponding to PIA. So if these field isolates, spray inoculation onto rice cultivar, which has a PIA, A means the isolate will show the A virulence, so it cannot infect. V means virulence, so it can infect. So, and this lower panel is sudden hybridization against the genome DNA of each isolate using the probe of ABR PIA. So if the fungus shows the phenotype of A virulence, there will be ABR-PIA. But it's showing virulence, no sequence of ABR-PIA. So all or nothing. There, in case of ABR-PIA, only the deletion is found among the field isolates of the without ABR-PIA. Okay. So the deletion will be the common mechanism for this ABR-PIA. So I would like to, at the time I thought that I want to solve the mechanism of the deletion. So in 2013, we could publish the proposal of the mechanism of the deletion. So from several slides after this, I'm gonna tell you how we could find the mechanism. Okay, so as I told you in the previous slide that if we use a resistant cultivar, for a while it's okay, but in several years, they will make mutation to make it overcome that resistance. That means resistance breakdown. So in this study, we used a parental strain and mutant. Okay, so this ENA168 is a field isolate of rice plus fungus, which has ABR-PIA. So this fungus has ABR-PIA, so it can infect this cultivar SHIN2. This doesn't have any R gene. So if they have, it has no R gene, it can infect, but it cannot infect the rice cultivar Aichi Asahi, which has PIA. So it's showing A variant. So this ENA168 M1951 is a mutant strain that is isolated by spray inoculation of this strain in high concentration of conidia onto Aichi Asahi. And eventually they makes the lesion. So we isolated the strain from there and we kept. So this mutation is spontaneous. We didn't do any induction of the mutation, just do the inoculation onto this Aichi Asahi. And it's showing the virulence. Okay, so this is the mutant strain coming from this ENA168. Okay, so what we first defined is the actual copy number of this ABR-PIA gene in the ENA168 parental strain and the mutant. Okay, so I was showing in the field isolates only the deletion will be there. And finally, we found that this parental strain ENA168 has 3 copies of the same ABR gene. 3 copies, not only 1. Okay, so if we digest the genomic DNA of ENA168 using EcoR1, it's showing 3 bands. That means 3 copies of DNA, the gene will be there. And mutant showing no copy. So a parent has 3 copies, a mutant lost everything. Okay, so it was fascinating a little bit. So 3 copies deletion at one time. Okay, and also we want to define these 3 copies of ABR-PIA is located on the same chromosome or not. So this is the separation of the chromosome using the pulse field gel electrophoresis. Okay, so this is ENA168. And if we probe with the ABR-PIA, only 1 band will appear. So this, okay, it's not so clear, but chromosome was separating. So only 1 signal we could detect. That means 3 copies of ABR-PIA are located on the same chromosome. Okay, so after that, we were trying to many sequencing, hybridization, or reverse PCR to clone the adjacent sequence to find the boundary. Okay, so we know now is that these 3 copies are lost, but we don't know from where to where is lost. Okay, so next step will define this boundary. And if the time permits, another 30 minutes, I will tell you about how we could define the boundary. But today the time was a little bit limited, so I will tell you about the results. Okay, so finally we found that 3 copies of PIA, and one of the copy of PIA, there will be a transposable element, DNA type transposable element named Okian. This element was found in my laboratory, but Okian means the funny girl in Japanese. But anyway, yes, this element we found. And from the information of here, okay, so this part was common in the mutant we found. So from here, this sequence, we found the same sequence in the mutant. So this part was still in the mutant. And then we checked this side in the mutant. And this side was totally different from here. But we found another place in the parent here. And we found another copy of Okian here. So after the sequencing of this Okian, we found that recombination between this purple transposable element and this green transposable element, these 2 are same transposable elements. So almost same sequence, but on a few point mutation were there. So if we look on this one transposable element, same transposable element in the mutant strain, it's showing the evidence of the recombination. So we could found the mechanism of the mutation of these 3 copies of every PIA deletion is recombination between this purple transposable element and this green transposable element to cut out this sequence in between those 2 transposable elements. Okay, so deletion was occurred due to the homologous recombination of the transposable elements. Okay, so transposable element is important in the rice plus fungus. I will introduce another achievement of my result, my laboratory. A similar work. It's about ABR-PIK. So this ABR gene was also in a 168, same wild type. And we could isolate the similar way as a mutant, the 5 mutant of ABR-PIK. So this ABR-PIK is also, I didn't tell you about the ABR-PIA was same, but it's very small gene, only 300 base pair. ABR-PIA only 286 base pair. It's a very small gene. So similar things we did. I will tell a little bit about this. So it takes a long time, but anyway, in a 168 has 1 copy of ABR-PIK, and all the mutant lost the ABR-PIK. Anyway, deletion happened in ABR-PIK. Okay, so after this was more complicated than ABR-PIA. So sorry, it's a very busy slide, but actually, we could publish this one in 2021, just 2 years before now. It takes also a long time. But okay, I will use the next slide to tell you more about we define this structure of the ABR-PIK and the sequence around the gene. These arrows in blue or green are all transposable elements. Okay, so ABR gene tend to locate in between the many transposable elements. Okay, and these transposable elements are key to makes the mutation. Okay, so this is the parental strain ABR-PIK locus, and this is the mutant locus corresponding to ABR-PIK. So it has half, maybe until here, until here, has a similar sequence as the parental strain, although they have translocation or transversion. And also it has some other sequence connected to the ABR-PIK locus. And we found those sequences in other clone of the genome of the parental strain. So okay, these two sequences are same as previous one. Okay. This one and this one. Okay, this one and this one. Okay, so the proposed mechanism of the ABR-PIK deletion is first, DNA will be broken in this POT2-like transposable element. Okay, it will be cut both of the strand. Okay, and then I located after cutting this shorter part at the bottom and the longer part at the above. Okay, and this very short part, but it can make the homologous recombination. And this recombinant fragment is this lower one. Okay, this upper part is this upper one in the other direction. Okay, and another homologous recombination happened here. And then this recombinant fragment here will be this one. And another one is from here to here, recombinant fragment. And I place here in the inverted position. And another recombination. Okay, almost same as the bottom one, right? Until here. So we need another fragment, DNA fragment in somewhere in the parental genome. So we found that from COSMID 26 clone recombination and another clone recombination. And finally, this part was lost, okay, including ABR-PIK. And it will become like the sequence we found in the mutant. So we can propose the mechanism of the ABR-PIK deletion only using the homologous recombination between the transposable elements. Okay, so many homologous recombination, but that is very powerful, and that's why, or maybe that is the point that these kind of ABR gene are located within the many different transposable elements. They can easily delete it. Okay, yeah. So, or maybe I will not tell you about that today, but by other researchers, it is proposed that these kind of ABR gene are regained from other strain, other isolates, or maybe same rice fungus, but other rice fungus by recombination. Okay, so I believe that this mechanism is one of the main mechanisms of the change of ABR genes in the rice fungus. Okay, so today I told about the ABR PIA or ABR PIK, but the deletion was found in other ABR genes in rice fungus. So I believe the deletion is most important mechanism of the ABR genes. So, and that deletion is caused by the homologous recombination. Okay, I will tell you a little bit about the recombination in the cell. So maybe some of you or most of you know the genome editing, right? So CRISPR-Cas9, okay, that the point of the CRISPR-Cas9 is cutting the correct position using the bacterial immune system, right? Okay, so but after cutting, it's the cell will do everything, right? So if we put after cutting, if we put some other sequence which has a homologous sequence, they will use that homologous sequence to change the gene, right? So if we don't put anything, the DNA will be deleted a little bit and jointed again. So cutting is very important. So after cutting, anyway, any kind of the organism has the way to repair the DNA. Okay, DNA repair is very, very important because DNA is very important for the organisms. Okay, so anyway, if the DNA is broken, any organism will try their best to repair. Okay, so after the DNA is cut in some position, they have two ways of the repair. One is a non-homologous end joining, just cut and just paste. I just connect each other. The other is homologous recombination, as I shown in the previous slide. Okay, so homologous recombination uses some other sequence. So this is red one is cut, and black one is other sequence. Using other sequence to repair this cut. Okay, when it occurs, it makes recombination. Okay, so that means the DNA cutting is very important. So I thought that in rice fungus, there should be many DNA damage or DNA double-strand break. That's why they make many recombination. That is my anticipation. Okay, so what I did is to count or to visualize that's DNA damage. Okay, DNA double-strand break. So one way of the visualization of the DSB double-strand break is to make foci here. I'm writing here. Foci is the multiple of focus. Focus. This is Latin. Focus, foci. Coccus, cocci. Right. So using a protein related to DNA damage repair, RAD51. So in this case, I think it's a human cell. Yeah. In the nucleus, we cannot see anything, but after double-strand break repair, if the RAD51 protein visualized using GFP or other method, it will make these dots, bright dots. That means the DNA break. Okay, so what we did in the rice fungus is fusing GFP protein to this homologous protein of RAD51, RHM51. Okay, so what we did is that this is a pressuring formation on the plastic. If the plastic is hydrophobic, rice plus fungus can make pressuring. They think, oh, it's rice leaf. Yeah, but this is very good because it's very simple to test. We don't have to prepare the host cell. Okay, so just put onto the plastic, they germinate and make pressuring like this. And if we observe under the fluorescent microscope, we could successfully see the bright dots. That means in only these, just germinate and make pressuring, they suffering from DNA break like this. Okay, and during the infection, okay, we actually use the rice cell. This is pressuring, and these are infection hyphae. So these are hyphae inside the rice cell. We could see some dots, and also in other cell, this is the hyphae in the cell showing the bright dots. So that means rice plus fungus suffering the DNA double-strand break during the infection. So to repair this double-strand break, if this double-strand break happening in the transposable element near the ABR gene, it will make the deletion of ABR gene. Okay, so that is, I think that is a very sophisticated mechanism to make mutation to the ABR genes. Okay, so this is the final slide of the part 1. Okay, so rice plus fungus will showing makes the infection, and at the same time they suffering many double-strand break, DNA break, and they should repair using homologous recombination. I have another data. I don't have the time to show, but the homologous recombination is more important for them. And they will repair the DNA damage by homologous recombination. And eventually, they may make the deletion of ABR genes. And if that is occurred on the resistant cultivar, that would be lucky for the rice plus fungus to expand, right, to overcome that resistance gene. Okay, so, okay, I will, oh, sorry, 大丈夫かな? I will change to the next topic, okay, mutation in the fungicide resistance. Okay, actually this part is not the make mutation, but this topic is related to spreading of the mutation. This topic is about the azoxystrobin or QOI fungicide. That QOI fungicides are increasing in the usage. At the same time, the resistant strain emerged and spread recently. Okay, so it's already resistant strain are found. So this inhibitor is related to mitochondria. So QOI, kinome outside inhibitors, that's inhibits mitochondrial respiration. So it will inhibit respiration and kill the fungi. So its point of the work is the mitochondria. Inside the mitochondria, there is a QO. Okay, so it has a cytochrome BC1 complex, but this QO will be inhibited by the QOI. The mutation of this against this QOI is found in the mitochondrial DNA of the cytochrome B gene, codon 143, by changing only one base, G to C. This is very common. not only the rice plus fungus, but any kind of the QOI resistant fungus strains have same mutation. Yes. So, and already this mutation is found in the field. There will be, if, of course, it's possible to stop making this mutation. But already it's spread and no other mutation are found. So there will be better to understand how this mutation is spreading in the field will be more important. Okay, so by the mutation, the QOI cannot bind to them. So what we interested in is that this mutation is happening in the mitochondria. So mitochondria is not the nuclei. So it's the inheritance is not like the other genes in the nuclei. Okay, so it's how many mitochondria will go to another cell, right? So rice plus fungus use conidia for the next infection. No conidia, no infection. So to spread this QOI resistance, conidia must have the QOI resistant mitochondria, not the hyphae. So from hyphae to conidia, how they transfer the mitochondria is very important. So to become QOI resistance, the mitochondria should be in the conidia. So we are thinking that how the rice plus fungus produces conidia. So if we observe on the plate culture, they will produce the aerial mycelium. At the tip of aerial mycelium, they will make the conidia like this way. Okay, first there will be a protuberance of the hyphae or conidiophore, and that will become conidia, and then another one will produce here and produce here like this. Another point I want to tell you is that about the mitochondria. Maybe this is new word for you, and that was new word for me when I was thinking about this. Is homoplasmy or heteroplasmy. So this tells the illustration about yeast. So in yeast, they makes zygotes. Okay, they are also ascomycetes. Okay, so if they have, each of them have different type of the mitochondria, after fusing, it has a different mitochondria altogether, okay? But after few generations, we can find the cells almost only one type of the mitochondria. So we call this is heteroplasmy. So which has a cell which has the different type of mitochondria. And then this type of cell, homoplasmy, only one type of mitochondria we call homoplasmy. We can think that these two things, okay, so conidia is very important, and homoplasmy and heteroplasmy to the spread of the resistance of QOI in the field. So if we use a QOI in the field, QOI resistance will spread very rapidly. I mean, the farmer says, or the other researchers on the experimental station says that if they found the resistance will spread in very short time. At the same time, if they stop the use of the QOI, that resistance will disappear very rapidly. So rapidly changing, okay? So I think this is related to homoplasmy during conidiation. So they need a conidia to spread the disease. And when they make conidia, they use a homoplasmy to make more and more a mutant mitochondria. And in case of no usage of QI, the disappear of the mutant. So what we did is that observation of the earlier time of the conidiation of the mitochondria, how they distribute the mitochondria to the conidia. And then we compare the change of wild type and mutant ratio before and after the conidiation. So how to visualize the mitochondria is use a TCA-related enzyme and fuse with GFP. So this enzyme will transcribe in the cytosol, but that protein will come to mitochondria. So after that, we could see that these green things are mitochondria. So how to observe the early conidiation? We made a filter paper, this shape, and this part will be inoculated with mycelium. And on the slide glass, put this filter paper and put humidity and cover with cover slip, and incubate and observe under the microscope. So, okay, this is already made one, and the new one will made from here. Okay, so you can see the mitochondria as a tubular. Okay, here, and next moment, 25 minutes. There will be a small new conidia will be produced, and tubular mitochondria is now coming into the conidia. Still tubular in 50 minutes. The 75 minutes, it start to dot, but it almost stop. This is separated. So in 100 minutes, it's already make a septum, and everything will be dot. So that means from the observation, first mitochondria will be coming as a kind of tubular to the new conidia. Not the many dots coming, only the tubular will come. So it makes showing the only a few type of the mitochondria coming to the new conidia. Okay, so in first it will come the tubular, and after making this septum, the mitochondria will become dots, and this might be showing the homoplasmy, but it's not clear. So what we did is actually counting the number of the copy of the gene or copy of the mitochondria during the conidiation. So using the qPCR. So there is a one gene base sequence difference between the wild type and mutant. So we have the system to amplify each of them. So using this, we made a qPCR system. So in case of wild type, we can amplify this part. And in case of resistant mutant, we will amplify this part. And this part of DNA will be quantified by the qPCR. So I will skip detail, but finally we could make the standard curve of number of the mitochondria in a DNA extraction. Okay, so the results. First, we checked using the field isolates of resistant and susceptible, 3 each from isolated from the real field in Hyogo or Akita Prefecture in Japan. And they are isolated in the same field, and we did the DNA SSR analysis to confirm that these 3 are very similar has DNA background. Okay, but they showing resistant and susceptible. And the count, after the count, resistance showing more mutant, susceptible showing more wild type. Okay, 100 times or 1,000 times, or sometimes more and more time difference in the copy of the mitochondria. So next we try to define the homoplasmy happening during conidation. So first we make a fuse of wild type and mutant to make heteroplasmy strain. And using that strain, first one way is to conidation, make conidia and count the number of the mitochondria. The other is no conidation, just make protoplast and count the number of the mitochondria. And this was compared with qPCR. So first we made a fuse, and in this number, fuse and number 3 showing almost 1-to-1 ratio of mutant and wild type. And what we did is a little bit complicated, but using that fuse strain, one is conidation as normal, that only agar make conidia and scrape, you get the conidia, single colony from single conidia, and do the looking on the how many copies of mitochondria. Okay, the other is liquid culture. During liquid culture, they will not produce the conidia. So in liquid culture, mycelium is used for the protoplast, and from protoplast we can make the colony, and we will do the similar thing, DNA extraction and no conidation, and just culture will be controlled. Okay, so if we do the conidation, so control is just parental culture. Okay? So 1-to-1, mutant and wild type. But after the conidation, wild type, most of the mitochondria in the conidia will be wild type. So this is actually almost homoplasmy. Okay. In case of without conidation, not 1-to-1, but a little bit tendency to mutant, but it's different from the conidation. Conidation changes 1-to-1-to-6 ratio, but without conidation 1-to-1 to 10-to-2 ratio. So it's by the conidation makes drastic increase of the wild type mitochondria in the next generation. Okay, so what we found here is that maybe this spreading of the QOI resistance is due to the homoplasmy during conidation. So we are now trying to found another way to clarify this using some mutants of the mitochondrial segregation, or this conidation experiment in another condition. So maybe the condition we used is favorable for the increase of wild type mitochondria. So maybe we can change the condition to put a little bit of the QI. Then maybe next generation will be most mutants. Okay, so this is the last slide I prepared. So takeaway message for young scientists, young in mind. Okay. So yeah, I talk maybe today the talk. What I talk in this morning was a little bit complicated, and maybe it's not easy to understand everything. The complicated and many things is that's the result of I was looking for. I used so many years to get results. I didn't tell you so much, but these days, okay, we have very strong machinery of the genetics, like NGS Okay, NGS will give so many information about the sequence. But in case of rice blast fungus, yeah, of course, NGS is very important, but not so strong, because you see that only the 20 or 30 kilobase range, we have many transposable elements of same sequence, right? So if we, okay, actually we are sending some cosmid clone, that means 40 kilobase or something, to the new NGS, not only the short version, but also longer one. But none of them give results as a one sequence. Okay, so and in the now is the time, not the time of genome sequence. Maybe you all know that all the sequence was done, but rice blast fungus, there should be unconnected part. Even now, they have 7 chromosomes, but the contact number in the rice blast genome sequence is not 7. It's more and more. So yeah, sometimes we need to do some classical method, but it gives this kind of results. Okay, so I think most important is enthusiasm. Okay, so nothing great was ever achieved without enthusiasm. So I think this phrase I love so much. I like this one. And I'm telling my students to be enthusiastic. Okay. Yeah. So this is end of my talk. Thank you very much.
Thank you. How to comment it? It's complicated, like you said. So first, from pressure to pericularia oryzae because of the resistant variety. That the first that I understand, and the second is about the fungicide application of fungicide. Will I mean the fungi will respond it with making the or change the mitochondria and become resistant to fungicide. And but when you are not applying the fungicide, then it's become wild type again. Oh. It's the for somebody. Do you wanna make a comment? Very complicated for me. Okay, so okay, please, please, if any question from you, also from our friends in the Zoom, joining in the Zoom, and also on YouTube. Okay, you first, and who else? Student, yeah, Mas Andra first, and then yeah, I could be for the student first, yeah, Pak Wid. Okay, Mas Andra.
Okay. So that the question is about the length of the DNA. Well, actually, we were looking for that, but unsuccessful. Maybe more than 1,000 kilobase, 1 mega or something. We were trying to cut the chromosome at the near the edge, we think, but they're too difficult. So I cannot say how long. Yeah, now. Maybe something like 1 megabase or something. Yeah. So it may happen. Actually, in that case, all the 3 copies of AVR PIA are located in the same chromosome. But we don't know the distance between each copy of the AVR PIA. So actually, I don't know the length of the deletion. That is, it's a good question that we couldn't solve yet. And also for the AVR PIK, even though we found the one side is the same as AVR PIK, but the other side of the deletion is on the same chromosome or not. We were trying, but it was not successful yet. So in that case, it's just not deletion, but might be the translocation. So it may make the recombination between the different chromosome. In that case, the story will be more complicated. But anyway, the homologous recombination in the transposable elements can explain how the AVR gene is deleted. Sorry, I couldn't answer to your question, but it's not clear now.
Thank you, Professor. So he is a master student, so could be it's an open research for his next PhD study. Okay. Another question from you?
My concern is, so what does the right leader is to do to overcome this? Because the resistance dependent on the fungicide is not really on the right.
Okay, good question. Yes, so the question is about or breeders how can manage or do in the breakdown. Okay, yes, this is very important thing, and in Japan, we have some kind of way using multi-line or multi-line system. That means, the Koshihikari or Sasanishiki is a very famous Japanese rice cultivar. Okay, Koshihikari originally doesn't have any R gene, but now they breed the Koshihikari with PIA, Koshihikari with PITA, Koshihikari with PIK, or any other rice resistance gene, and they mix them and plant. So as a rice, as a taste, it's Koshihikari, but it has each different R gene. And okay, if the one of the rice plus fungus change do the mutation to overcome one R gene, but they cannot go another. So as a total, they have a very effective to control the rice plus, and not the totally breakdown will occur. Okay, so that is one way. Yeah. So now a similar one was used for the other rice cultivars in Japan. That is one way. Yes. Yeah.
Okay, so multi-line resistant. So you plant a lot of kind of resistant from the plant, from the rice. I mean, so many different resistant kind of resistant in eggs and plant it in this one area.
Yes.
So it can be like a buffer for the pathogens.
Yes. Yes, that's right.
We know only from the theory. You see, that's why we are here. Okay, and another could be the similar question in the Zoom meeting. Is sorry, I will read it for you from Nurfianto Giar Pangidum. Two questions. How can the AVR linkage of Magna Forte Orize recognize rice are genes be applied to breeding rice blast resistant varieties? I think.
Cari tiket pesawat.
And what are the appropriate breeding method for this approach? And the second one, what are the strategies to ensure the resistance of rice varieties to blast disease or other pests and diseases is not broken in the field? Yeah.
Okay. So how can the ABRG engage of a recognized rice blast be applied to breeding? Yes. Okay. So yeah. Well, I'm not a breeder, so I cannot tell so much about this. But yeah, maybe one answer is that doing this kind of research, we can know. I believe that from my results, most of ABR gene can be mutated by the homologous recombination. But if we check all the ABR genes, maybe we can find one ABR gene that is very stable, which is located in the normal region without the transpose of elements. And maybe that ABR gene will not mutate it easily. So if we find that and use our gene against that ABR gene, that resistance will be more stable. Yeah. Or another way is that we know that mutation can happen by homologous recombination. So this is not the breeding strategy, but the other way is that some agrochemicals can inhibit the DNA repair of the rice blast fungus. Homologous recombination may make it slow, the mutation. Okay, so we tested some mutant of rice blast fungus, which lacks the DNA recombination enzyme. It will grow very slowly, and in a infection trial, they cannot infect rice. So the DNA recombination will be a target of new agrochemicals. Yeah.
Okay. Thank you, Prof. Other question from student? So, mutation happen on the Pediculare orizae. It's because of stress from the plants, plant resistant. If you saw that it's because of the ROS, reactive oxygen species, make the fungi chains and lost the FER genes. Please, Pak Widodo.
It is very complex what happened inside. I almost only a few, maybe only 2% understand about this what happened over there, and I almost know what happened outside. I mean, phenotypic, for example, breakdown of the cultivar, resistant cultivar or something like that. My question is, because between the pathogen and also rice or host is interaction, is it depend on the how frequent they interact with each other? For example, is it different between 4 season countries and tropical countries? In tropical countries, almost evergreen in the landscape view. Is it also depend on the, I mean, how frequent they interact. For example, in four-season countries, maybe it's a little bit short than tropical countries that rice is always in the fields, like that. It's very interesting thing, and it is too late for me to start with this with this item. But I hagiasang ni watasimasu. Domo arigatou.
Okay, thank you very much. So yeah, I think it's not too late. But yes, I think that it might be different between countries. In tropical countries, I think more generation will be made in a shorter time. So I think more faster the fungi will make mutation. Another thing is the temperature. So maybe higher temperature makes more stress to the fungi, or the more oxidative stress. More respiration means more oxidative stress, and that might have some effect, influence to the ratio, or that's how happening into the mutation. Yeah, thank you very much.
Yeah. Thank you, Prof. As we know, with last many years ago, we think that the Pericularia oryza is only on Padigogo, what is a dry upland rice, but now it's becoming the also, and could be about 4 or 5 years, no, since okay. Yeah, but before that is and we think about mutation also. Yeah, but no research yet about that. Or because it's upland rice first, and then now become a big problem also in the lowland rice. Okay, we hope Hagia will tell us something about that later on. Okay, any question from? Okay. A student, are there any? Please. Okay.
Thank you so much, Seno Sensei. Konnichiwa. It's really great opportunity to join this lecture, but it confused me so much. It's really complicated for me, but because I'm a young scientist, like you said, about the enthusiasm, so I want to ask, is it any possibility to recover the old gene mutation to the field? Like, is it any possibility?
Your question is the possibility of the mutation recover in the field? Yeah,
to recover the old gene mutation, the old genes.
Yes, I think so. Yeah, there is another research that about the ABR gene. The location of ABR gene in the rice plus fungus are different between the isolates and the different chromosome. Each isolate which has ABR gene has on the different chromosome. That means the story is that they will lost them and then regain to the other part. So not the same part, original part, they regain the gene. So there is one proposal that the rice plus fungus are losing and regain many times the ABR genes. That which means that you're correct that it may possible. And on the field, like I said that koshihikari in Japan, koshihikari doesn't have any rice resistance gene. So as a population, when we introduce koshihikari to the rice field, the blast fungus we found from them has many ABR gene. But before that, if we planted other rice cultivar with R gene, some of them are showing the no ABR gene. So in the field also we observe the regain of or recover of the ABR gene. And also for the agrochemicals, as I shown in the slide, that it is in the field we observe that it's easy to turn to wild type as a phenotype. Yeah? But what I believe is that it's a phenotype is wild type, but it may still have the 1 or 2 copies of mutant. You know? So when they apply the QOI, they will increase the mutant copy and they will change as a phenotype. I think that will be I believe that will be the truth. Yeah.
Selamat pagi. My name is Aslan Ashad. I'm from Pakistan, student of plant breeding and biotechnology department. Thank you, Professor, for coming and giving this special line. Nothing great was ever achieved without enthusiasm. As for me, I'm student of genetics. So thinking about these genes and deletions and mutations, sometimes it's like understanding, sometimes it's going like overhead. and as you said, like NGS is nowadays, everyone is having like a deep concern about NGS. So I was so enthusiasm like about studying about NGS because it's a new technology, but right now I'm studying, so I just want some more motivation to graduate. So my question is about like, I was late, so I read one slide. It was about fungicides. They are least resistant and environmental, somehow like friendly. I saw some slide. So mutations are always harmful. So do they lead to, how to say, host specificity and how to say?
Maybe the list of the fungicide. Correct?
Yeah, here resistance strain emerged.
Yeah.
Popular and important.
So
risk for environment.
This one, eh?
Yeah.
Okay. Yes.
Thank you.
Yeah. Why is it? Your question is.
Yeah.
Okay. So yeah. Yes. So this one will affect the respiration of fungi. This is actually this compound is also made by a kind of fungi. So it's produced by organisms, and only this compound will inhibit one of the pathway in the respiration. So usually this one will not affect the respiration of normal plant or other species. Only when the rice blast fungus or the fungus in the plant cell, the fungus can use only one pathway in this respiratory chain. So they usually have another pathway, bypass here. But in in plant, I mean, if they growing inside the plant, they can only use this pathway. So this QOI fungicide are only effective for the fungi which growing inside the plant. So it's not affect the respiration in the other plant or other usual area which organisms living in air. Okay, yeah. So, and I think it's a natural compound, so it can be digested by the other microorganisms. It is coming to the soil. Yeah, I think that's why it's called like this. No risk for environment.
Okay, thank you. Other question? Okay.
Thank you very much, Ibu Evi. I am Nurholis, Professor Sone, and I am Hagia's friend. Thank you very much for sharing the knowledge about the mutation. We know that there are several types of mutation, like substitution, insertion, and deletion. And in your study, you said that deletion is the main mutation in Pyrrhucularia oryzae. And my question is there specific reason or specific factor that influence why the type of mutation in Pyrrhucularia oryzae is the deletion, not insertion or substitution? Thank you.
Yes, good question. Well, it's a good question and difficult question to answer. Yes, what we found is that the location of the ABL gene we found is surrounding by the many transposable elements. So I got idea that the homologous recombination is one of the main way to make the deletion of ABL genes. And the other thing is that I said that many transposable elements in the genome, but it's not easy to detect the translocation of each transposable element itself. So yeah, actually we can find some insertion to the ABL gene of the transposable elements, but I think the activity of the transposable elements are not so high because it's already too many copies of each transposable elements in the genome. For example, what I shown in my slides, those transposable elements are more than 50 or maybe 100 copies in the genome. So if they turn on everything at once, it will be very big risk for them. So they stopped using to cut and paste or copy and paste way, but they utilize that as a point of make the recombination. That is another my guess, but actually I don't know. Yeah, yes, but what we observe is the DNA is actually broken in the cell, and if we inhibit or cut the recombination repair by making the mutant, the rice blast fungus will grow. The growth was very slow, and they cannot make the pathogenicity. So the repairing is very important. I observed. So from that, I'm thinking that homologous recombination is the most important. But yeah, not directly evidence. I have not yet found. Sorry.
Thank you. Bahasa Indonesia juga boleh. Okay.
Good morning, Sensei. My name is Marlina Puspitasari. I'm from plant pathology, doctoral program. My question is about mutation in AVR genes, either AVR-PA or AVR-PAK. Is it can affect the morphology of the fungi or maybe the symptom in the plant? That's my first question. And the second, in the field, how we can differentiate between the mutant one and the original one just from the symptom? That's my question. Thank you.
Yes. For the usual, for example, culture, we cannot distinguish. No morphological difference. Only when we do the inoculation assay onto the rice, which has the R gene, or which doesn't have R gene, we can distinguish them. It's not easy, actually. So, and the how to define it's a mutant? That's a very good question. And in our case, we use in a greenhouse assay using our single, the cultivar and the single strain, and just inoculate and isolate. So we can believe this is a mutant. But if we just isolate from the field, we cannot say it's mutant, because just maybe it's just lost the. we cannot find parent, right? So yeah, what we believe, just one way is to believe, but the other evidence is that okay, for example, in case of rice plus fungus, we can do the SSR, short sequence repeat fingerprinting, or some repetitive sequence, MGR fingerprinting, to compare whole genome. And if they're showing very similar, maybe we can believe this is mutant. Yeah.
So no difference, even the color that they produce, I mean, like apicularia oryza will be darker. It's not different also, or?
Not different.
Oh, okay.
Not different.
Thank you. And is it okay now? You know? Any question? Bahasa Indonesia boleh. Miku in Japan is okay. No chat? Okay, Pak Gian the first.
Okay. Okay, thank you, Bu Evi and Sone Sensei. Very interesting. From your presentation, I can imagine how smart the microorganism is.
Yeah.
Very, very smart. Some question already presented by student or others, but the important, the most interesting is how the interaction between plant and the pathogen itself. We know the concept of arm race between plant and also pathogen. In your research, is any observation also for the plant itself when they infected by the pathogen, also how the pathogen decides to make mutation to activate transposon and so on and so on, both for the AVR and also for the fungicide resistance. It is very, very interesting. Thank you.
Yes. So yeah, actually, yeah, I have same feeling. It's very interesting, but I don't know which is first or no. You mean that the cock will be first or egg go first, right? And it's the interaction. And I think one interesting thing is that Pilicaria, we have two strains, two species. One is Pilicaria grisea, the one is Pilicaria oryzae. So Pilicaria grisea is a pathogen of the normal grass. It's just not the cultivated ones. Pilicaria oryzae is the rice or millet or such kind of we cultivate ones. The number of transposable elements are different between Piliculariae grisea and Piliculariae oryzae. Piliculariae oryzae has so many different kinds, so many copies of transport topological elements. And if we look on the chromosome using the pulse field gel electrophoresis, we can see the band. And Piliculariae oryzae has very, very different lengths of the chromosome in each strain. We cannot believe this is the same species. So what I think is that rice plus fungus Piliculariae oryzae has, in a way of evolution, they gain the ability to make mutation by putting the many transposable elements and repairing the DNA using the recombinatory repair. So I think that is very important. For that, the rice is very important. Okay? So they will infect rice, and then maybe on the rice will be cultivated by humans. So human will, they try to control by changing the cultivar or something for long history. So that makes the Piliculariae oryzae more and more easier to make mutations. So that, as you said, it's a interaction between plant and the pathogenic fungi makes more and more sophisticated. Yeah, thank you very much.
Thank you, Sensei. Any question? We still have 5 minutes for question. Bahasa Indonesia boleh kok. In bahasa, please.
Okay, thank you very much, Sensei, for your nice lecture. I think we learned a lot from here. I just learned that you've been studying or doing research with the blast fungi for a long time. And here, I think most of us as a plant pathologist understand that blast fungi is the most diverse fungi, one examples of the most diverse fungi in the world. And we just want to know, in Japan, for example, how many races of blast fungi exist now? Because, around the world, people just talking about new races coming every times, and maybe you can share with us, the latest study about the races of Piliculariae in Japan. And then another question is about the isolate INA 168 that you did the mutation study. Why did you pick in the first place for this isolate in your study and not other isolates, something like that? Yeah.
Right, good question. But the first part, well, number of races, I don't know. I don't know, but yes, to tell the truth, that we do have the rice blast in Japan. This, of course, every year, but not the large area was infected. Now we are controlling, so under the it's quite a low level. So actually, I don't know the data of the current race. So, well, but I mean that if we introduce a new R gene, the new race will come. But now in Japan, we don't increase the new R gene. So just use the 9 or 10 R gene. And as I explained that as a multi-locus, multi-line, so they will control. So and they, yeah, each and combining the such R gene like PI something, that is a strong one, right? The another one, some more weaker or horizontal power R gene will be there, and then the combination of that are effective. So I think that the number of races in Japan are not increasing now. The other question is that INA 168. INA 168 is actually one of the strain that used for the first we make pathogenic race in Japan. Okay, so it's the INA 168 race 101 in Japan. That means only one ABR gene is lost. 101. 101 is yes. Yeah. So in other words, it has most of the ABR gene. So we can use that for to make mutations of ABR gene. It's better to use the parental strain as much as ABR gene will be there already. So we can delete from there. Okay, and yeah, easy to propagate. Yeah, and stable. I mean, the some of the rice plus fungus which newly isolated from the field, it's okay. It grow well in the beginning, but sometimes it's lost the growth or the spore production. But INA 168 is already isolated in 1950s, and they used for many works in Japan. So it's very stable, and it's a kind of the laboratory strain at the time already. Yeah, that is another reason.
Very interesting. Yeah, so multi-line is very effective if we hear from your comment. Okay, so then we disturb the environment with new R genes variety, then the pathogens will come up with new lost AVR genes again. Okay. One more? No more? Is it enough? Okay. Thank you very much, Professor. It's very interesting and make us evaluate what we have learned from our courses, many courses, and then we now like now we heard about multi-line. For me, it's very interesting because in Indonesia, when we have a new variety, then all farmers will plant the same varieties, and then yeah, like the professor said, something will come up because of that. Okay, so thank you so much for Andra, Bu Sinto, Pak Nurfianto, Pak Widodo, kemudian Mbak Jahra, Mr. Arslan, and Pak Nurholis, Bu Marlina, Pak Gianto, Bu Asti for the comment and make our seminar very interesting. We hope we learn a lot more from this discussion, and then for writing, so you have an idea what to write on your papers, even if it's not belong to this, but then oh, I must think about this one, I must learn about this one before continuing submit my paper and so on. Okay.
They're waiting for the exam.
The exam? Okay. Oh, yeah. We give it. It's depend on the lecture for each courses. Pathogenesis, pesticides. Okay. So again, thank you so much for your presentation, and we have a small certificate. May I invite who first? Bu Asti first to give the certificate for Sensei. Please, please stand up here, please.
Okay, Sensei, on behalf of the department, we would like to present the certificate, and thank you very much for a very nice lecture. Thank you.
Thank you so much, Professor. And Pak Widodo, would you like to come here? We have small things for Professor. Again, Prof, please.
IPB からdari mana? Oh, from Department of Plant Protection. そうだ、これ、おかしい。お土産。there's a bonsai. Okay. Thank you. ありがとうございます。
Terima kasih, Bapak. Okay, and this is the last of our seminar. We will take a picture with you. Now the era of picture. Is it okay? The student will take a picture with you. Is it okay? Okay, so come up, come here. Could be we change to the title, the first page. The first page of your presentation. Yeah, come here, come here. The student first, come, please. Yeah, student first.
Oh, thank you.
Merapat aja, merapat.
Badannya dimilihkan. Picture. Yeah, so we have to take pictures.
You have to do the end of the one.
Oke, Australia, Europe, oke.
Saya mutation dari Jepang. Pak juga mutation di sana.
Ini kamera siapa? And also from Japan.
Oh, I see.
Terima kasih semua, terima kasih sudah hadir, sampai ketemu. Terima kasih, terima kasih.